Hello everyone, I'm Romane Junker and I'm currently doing my PhD in the Maillage Laboratory at Paris-Saclay University. And my PhD focuses on bacterial communities from lacto-fermented vegetables. So I'm really happy to have the opportunity to talk to you about my work on integrating metagenetic datasets through microbial association networks to compare microbial communities from lacto-fermented vegetables. I don't know if you're familiar with lacto-fermented vegetables, but it's food such as kimchi or sauerkraut. There is currently a renewed interest in the bacterial community of fermented vegetables, since this type of food is at the heart of sustainability and health issues. So lacto-fermented vegetables are the product of a fairly well-described spontaneous fermentation by lactic acid bacteria. However, the microbial community succession has received little attention in lacto-fermented vegetables. Thanks to the progress of sequencing technology and open data policies, there are numerous microbiome datasets on food ecosystems that are available and reusable, so meta-analysis can be performed in order to better understand the microbial communities. But it raises many challenges related to data reusability, integration, and interpretation. So questions such as how should independent datasets be processed? How can the results of an integrative analysis can be visualized? And microbial association networks are a promising approach for performing a meta-analysis, but it raises further questions. How can networks infer from different independent datasets and studies be compared? Is it possible to identify a meaningful core network? And does the network structure reflect the structure of the bacterial communities? So in this study, we present an integrative bioinformatics approach through association networks to integrate public amplicon datasets monitoring lacto-fermentation in vegetables. First, we acquire datasets from NCBI SRA, Magnify, and Food Microbionet, and we selected those with at least 2 time points to carry out temporal monitoring, more than 10 samples in the study to compute association metrics, an associated publication to have access to metadata, otherwise it's impossible to reuse the data, and the same RNA region sequence to compare the ASV, donc amplicon sequence variants, the exact sequences. Then we obtain a great number of 10 datasets that were reusable from 5 different vegetables: carrot, cucumber, red pepper, cabbage, and radish. And they include between 11 and 206 samples and 32 unsynchronized time points, as you can see here. With those datasets, we process the sequences in data by study by using the data2 pipeline to do ASV inference and filtering the ASV on abundance and prevalence. And then with each ASV count table, we infer an association network by study by combining several metrics to obtain co-presence with the Jacquard distance and co-abundance with the Pearson and Spearman correlations and proportionality on cellular transform abundances. For instance, here you can see the result for one study. So on the left, it's the classic bar plot showing relative abundance. So the samples are here. They are ordered by sampling times. There are 18 samples and 6 different time points. And here is the network. So each node is an ASV. The color represents the taxonomic order. So in violet, it's Enterobacteriales, blue Lactobacillus, and yellow Pseudomonades. And the size of the node is the maximum relative abundance. There is an edge between 2 nodes if at least one metric found a positive association, and the thickness of the edge represents the number of metrics. Moreover, we reorder the node here with the x-axis, which represents the weighted mean edge, which is the edge of the samples where the ASV is found, weighted by the relative abundance. For instance, this ASV is mainly found in samples of 30 days. Okay, so what can we see here? First, there are kind of two patterns in this network, a high diversity here of ASV observed in the initial microbiota here. And on the second part, you can see lactobacillus and some enterobacteriales observed mostly in the final microbiota. So the network really reflects the dynamic evolution of the microbial community during the fermentation of this study. Once this is done, we can compare and integrate the network by building a core network with edges found in at least 3 networks and clustering all the networks using the multiplex stochastic block model clustering. So do independent studies display a similar pattern? Here you can see the core network built from more than 500 samples and 11 networks. And you can see a common temporal succession with a shift from the initial population present in the vegetable microbiota to lactobacillus in blue through enterobacteriales. Moreover, when we look at the clustering results, we can see that there are 10 different ASV clusters, and the cluster 5 here in bright green highlights an association between enterobacteriales and lactobacillus. So that might represent an early fermentation core consortium with a potential application of the fermentation initiation. To conclude, the public dataset integration through ASV association network provide additional value compared to independent study analysis. For instance, here we found that enterobacteriales presence at the beginning of the fermentation is a recurring pattern that raises the question of their ecological role in the fermentation of vegetables. What's next? So this is a first valuable approach that can be improved with the robustness measurements of association, especially in the core network. And the biological interpretation of this work is limited by the number of available datasets and metabarcoding technology. That highlights the need for complementary time series functional studies. Thank you very much to all my colleagues, and thank you for your attention.